my channel Biology Access. Okay, so today we are going to learn Unit 6 Core Practical 1. Okay, so let's get started. The first practical is investigate the effect of light intensity, light wavelength, temperature, and availability of carbon dioxide on the rate of photosynthesis using a suitable aquatic plant. Okay, so before starting the experiment, I just want to tell a few things about Unit 6. It is that in Unit 6, we need to write, in the exam, we need to write all the experiments in detail. So, in minor detail, with all the details, okay. So, if you do an experiment, you have to, like, if you write an experiment in the exam, you need to give all the factors in it, all the things which is affecting the experiment, the dependent variable, the independent variable, which is the most important thing of the experiment, the complete method, the procedure to do it with details, with all the tiny details in it. Okay, it's quite tough to remember in the first place, but if you keep trying it more and more, it will become a lot easier. Okay, so today we'll be learning about the effect of light intensity and light wavelength on the rate of photosynthesis, okay, by using an aquatic plant. So here the independent variable, which is the independent variable, light intensity and light wavelength, both are independent variable. And which is the dependent variable over here? The dependent variable is the rate of photosynthesis. Okay, the dependent variable is the rate of photosynthesis. So what is meant by independent variable and dependent variable? Independent variable is the variable with which we can change. Okay, by changing the factor of independent variable, we get the result, and the result is the dependent variable. So here how we can investigate the light intensity, by doing an experiment with different light intensity, we can figure out the effect of light intensity, okay, on the rate of photosynthesis. So before starting, I just want to I just want to make you understand the whole procedure first, and then I'll be giving in the notes. Okay, here the independent variable is light wavelength, light intensity, temperature, and availability of carbon dioxide, whereas the dependent variable is the rate of photosynthesis. So in today's video we will be learning about only light intensity and light wavelength. Okay, so let's get started. So during this experiment, what we have to do is that we need to first imagine the whole experiment in our head, that it should be like that we are actually doing the experiment. Okay, and just memorizing the experiment will not make it easier for you. If you understand the experiment, if you know how to do it, it will be a lot easier. Okay, so how we can like do this investigation, how we can measure the light intensity. Okay, we can measure how we can measure the rate of photosynthesis. Okay, we can measure the rate of photosynthesis by the production of oxygen. Okay, isn't it? The rate of photosynthesis increases when the amount of oxygen increases. Okay, so this is the variable what we need to find. Okay, so how we can find how we can like calculate our result? It's like we can figure out the volume of oxygen, right? Then we can compare with each of the variables, like in which variable it has higher amount of oxygen production. Okay, and how we can give independent variable, like in case of light intensity we can give bulb, the light bulb with different intensities, and in case of light wavelength we can give lights with different color filters. Okay, to do this experiment we need to have an aquatic plant where the procedure will be At first, we'll be discussing about how we can control light intensity and how we can measure the rate of photosynthesis. Okay, so to do this, at first we need to collect a piece of pond bed approximately of 10 centimeter in a large beaker of water, and then we'll be placing it in a beaker and removing any excess air bubble present in it, okay, by just running a finger or a thumb over it. Okay, then we will cover one side of the beaker with aluminum foil so that light can only enter from the one side to control the variable or make the procedure the experiment more reliable. And to do this, like in Unit-6 experiment, you need to write the experiment in complete detail, okay, to gain complete mark. And all the details is really important, even if it's the size of the plant you take, or any other variables like the instruments you use in the experiment. You need to name all these instruments to get a complete mark on Unit-6. Okay, and then what we'll do is that we will add a half spatula of hydrogen carbonate. Due to it's an experiment inside the water, so we need so the plants need carbon for the photosynthesis process, right? So the sodium hydrogen carbonate works when it gives the carbon to the plant, aquatic plant. Okay, and then we will fill the capillary tubing of the photosynthesis with water to start the experiment. Okay, and then we will continue with the experiment. Let me just show you a figure first to make it a lot easier, to make it more understandable. Okay, so this is a typical figure of the experiment. Like, it's a beaker where a thermometer is given to measure the temperature, and one side will be covered with aluminum foil so that light can enter from the one side only, okay, not from the other side. Here is the capillary tubing. The syringe is to take the oxygen amount, to count the oxygen amount. We can also like count the bubbles it forms, but that is less reliable because it does not show the right amount of oxygen produced. Okay, it just gives an estimate idea. Okay, but if we have the volume of oxygen, that is more reliable. And we have the pond weight, the skull, and this is the capillary tubing. Okay, so let me just go with the theory first. Okay, so this is the procedure. Like, at first, place a piece of pond weight approximately 10 centimeter long in a large beaker of water. Remove any bubbles by gently running a finger and thumb over the surface of the pond weight under the water. Cover one side of the beaker with aluminum foil so that light can only enter the beaker from the other side. Add half a spatula of sodium hydrogen carbonate to the water and leave for 5 minutes. And it works as a carbon source. Okay. Position the bench lamp 10 centimeter from the beaker. Allow the pond weight to adjust for 5 minutes. Okay, this is the procedure, and to gain complete mark, you need to write exactly the experiment, like all the details, all the instruments you use in it, like all the little, also even the little details is important for Unit-6, to be honest, to gain a complete mark. Fill the capillary tubing of the photosynthesis with water. Place the funnel end of the tubing in the beaker of water and position the pond weight with the cut end at the top of the funnel opening. A paper clip attached to the opposite end of the pond weight can help to weight it in the correct position. Like, the pond weight will be in a upward direction, like the roots will be in the up and the leaves will be in the down, so that when oxygen leaves from the roots it goes up the tubing. Okay. As bubbles of oxygen began to form and pass into the capillary tubing, start the stop clock. After a suitable time, use the syringe to draw any oxygen produced into the capillary tubing. Record the volume of the gas produced. Okay. Use the syringe to refill the capillary tubing, then begin recording again. Use the same pond weight and apparatus. Repeat step 7 and 8 with the lamp at different distances from the beaker. Take repeat measurements for each distance. So what it says that when 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 this pond weight releases the oxygen, it travels from here to this side. Okay, it travels like this. Okay, so any oxygen molecules over here will be drawn back through the syringe. Okay, and water will be supplied again. Okay, and we can count the volume of the oxygen over there to for measuring the dependent variable. Okay, and this is how the experiment done, and this experiment is repeated several times with different light intensities to make the experiment more reliable. Okay, so now let's move on to the next one, which is to investigate the effect of light wavelength. Okay, so let's go with the theory first. Place a piece of pond weight approximately 10 cm long in a long beaker of water. Remove any bubbles by gently running a finger and thumb over the surface of the pond weight under the water. As I said before, cover one side of the beaker with aluminum foil so that light can only enter the beaker from the other side. Add half a spatula of sodium hydrogen carbonate to the water in the beaker and leave for 5 minutes. Why do we leave it for 5 minutes? So that the plant can adjust over in that atmosphere and gives a reliable result. Position the bench lamp close to the beaker with a colorless filter. It's the colorless filter between the lamp and the beaker. This will be a white light control. Allow the pond weight to adjust for 5 minutes. Okay, we give the plant always to adjust for 5 minutes after installing a after installing a new instrument to it, so that the plant can get used to its new environment and we do not get any unreliable result. Okay, then fill the capillary tubing of the photosynthesis with water. Place the funnel end of the tubing in the beaker of the water and position the pond weight with the cut end at the top of the funnel opening. A paperclip attached to the opposite end of the pond weight can help to weight it in the correct position. Same as before, we need to place the pond weight upside down so that oxygen can go out from the roots to the capillary where oxygen number is counted. Okay, or the volume is measured. As bubbles of the oxygen begins to form and pass into the capillary tubing, start the stop clock after a suitable time. Use the syringe to draw any oxygen produced into the capillary tubing. Record the volume of the gas produced. Okay. Replace the filter with a filter of a different color and leave for 5 minutes. Okay. So what we have to do is that we need to replace, as I said before, that a colorless filter is placed. Okay, so that we can measure so that we can know the effect of light wavelength. Then this colorless filter is changed with different colors so that we can do the same experiment with different light wavelength and find the best one among them. Okay, so then what we have to do is that we need to repeat this experiment again from the steps 5 to 8 with different color filters. Okay, as I said before, with different color filters, and then we can measure the oxygen volume and find the best among it. Okay, let me just explain it once again. This is the experiment, and this is the this is the apparatus, this is the beaker, And a thermometer is used to check the temperature control, because we want the temperature to be same throughout the experiment, for all the experiments. Okay, because if temperature gets changed, the rate of photosynthesis can get altered. Okay, and an aluminium foil, so that light can enter from the one side only. Okay, and we need to give same amount of sodium, hydrogen, carbon to all the beakers, like to all the experiments we do, so that the carbon amount is always same. Okay, we need to give except for the independent variable, the other variable must be controlled to make the experiment more reliable. All right, so thank you for today.